So this is the data collection for part 1. We have already put the different acid concentrations in, and you can see those marked there. We're going to be adding 2 indicators, methyl orange and orange 4. And what you want to focus on are the colors that are produced. And Miranda's going to start adding indicators.
Okay. So just in this top right here.
Yep, which one is that?
Methyl.
Yep, so all the way across the top, like two
right.
drops is probably plenty. Oh, that's working pretty nicely.
Okay. Oh, perfect.
Yeah.
All right. There's one.
So notice that obviously all the colors aren't the same. You have for sure 3 different colors. You've got the yellowy, orangey color, and then it starts going into pinkies and things.
All right.
And here's the second indicator.
Ooh, that's so great. So, first one, it's like pink.
So we are now going to, so these are your standards from part 1. We're going to use these colors and compare them to the weak acids. So HCl is a strong acid, so it 100% dissociates. That means that whatever the concentration of the acid is the same as the concentration of the H+ ion, because you have 100% dissociation. In part 2, we're going to use weak acids and bases. Those concentrations of H+ are going to be less than the acid concentration because weak acids don't dissociate 100%. So the idea is to use the colors from part 1 in order to figure out what the H+ concentration is in part 2. So we'll pause the video and we will restart it when we have the second set up for you. So we are now ready for part 2. All of the weak acids are in the test tubes, and Miranda is going to add the 2 indicators just like for part 1.
Wait, this methyl orange. Okay. All right. Okay. And then we've got orange 4.
Mm-hmm.
Okay.
So now what we have to do is we have to compare these colors to our standards from part 1. So we're going to have to pull back a little bit.
We could zoom out.
Yeah.
Oh, okay. And then
So
here, if we like, we can move this.
did you, are we still going or did you stop?
Yeah.
We're still going? Okay.
We could stop it.
Let's stop and we'll stop it and get it set up. All right, so now we're going to start comparing the colors of the weak acids to the colors of the strong acids.
All right. This is methyl orange, 0.1 acetic acid, 0.1 molar.
Okay. So the 4 on the left are the methyl orange, so those are the only 4 we have to worry about. So as you, I'm gonna actually get a little closer, as you compare, you can see that it's probably in between the second and the third. So it's not quite red as the third one, but it's definitely more red than the second one. So we're going to call that 0.005 because it's in between the two. Okay? Miranda just wrote that down. So now we'll do the orange 4.
Orange 4. So this is acetic acid, 0.1 molar with orange 4.
So again, little less yellow than the first one or the second one, but definitely not as orange as the third one. So we'll call it 0.005 again. So then we're going to be quiet and let you guys decide what the rest of the colors are.
All right. This is methyl orange, 1 molar acetic acid.
Put it back there a little bit longer. Let's make sure they get a chance to compare. So you're comparing it to the 4 on the left. Where do you think it falls in terms of color? Remember that the concentrations are going from 0.0001 to 0.1.
And this is acetic acid, 1 molar with orange 4. This is 0.1 molar H3PO4 with methyl orange. This is 0.1 molar H3PO4 with orange 4. This is H3PO4 1.0 molar with methyl orange. And this is H3PO4 1.0 molar with orange 4.
Okay, all right, so what you want to do is use those color comparisons. These are the known concentrations. You know what the H plus concentration is for the HCl's. You're trying to figure out what the H plus concentration is for the acetic acids and the phosphoric acids. So that's your task. Match up the colors, figure out what the H plus concentration is, and then you're going to use those to figure out what the Ka's for phosphoric and acetic acid are. And I want to thank Miranda for helping out.